Swayam Prabha. Digital India. Educated India.
Dear students, hope you all are doing well. Welcome to another very exciting session of the course, Soft Tissue Characterization and Applications. So today we are going to talk about a very interesting topic which is on clinical testing with tissues. So what all clinical guidelines and other ethical requirements are needed, like to test tissues like which are either cadaveric or animal based, right? So that's one of the very important aspect of experiments and making sure that all our soft tissue mechanics based studies can be validated with realistic testing, right? So in order to do that, like, we need to make sure we are all conforming with the clinical testing norms, and we'll also be studying about some of the case studies with this clinical testing as we go further, right? So let's get started. Okay, so first of all, from a clinical testing standpoint, which is not only confined to tissues, it is a broader set of rules, regulations, and practices which have come along over the years. And this not only focuses on the tissues, but also biomaterials, medical devices, anything which has to do with the human body, be it dead or alive, right? Like, so anything which is humans or animals involved, that's where clinical testing norms and this term in general, like clinical testing, is very popular, right? So coming back to this, so what is clinical testing exactly? So there is a clinical problem, right? Typically that's where it starts from. So we do not just randomly start with certain tests, right? Like, we have to define that why we would like to run a certain test. मुझे बस 10 सेकंड का टाइम लीजिए, I just need to respond. Recording হচ্ছে, আমি busy আছি, इसको जरा skip करा देना. Okay, so this is what we do. So basically what happens is you start with an idea, and there is a design of the system to make sure that you're attending to a certain clinical problem, right? And then finally you do some validation which involves certain soft tissues from animals or the human models and so forth, right? So this includes like in vitro animal models, ex vivo models, clinical testing and studies like on human trials and things like that, right? So this is this broader scheme of things for clinical testing. So within clinical testing, biomaterials and medical devices constitute an extremely diverse and heterogeneous categories of items, right? So so you cannot define that this specific thing is biomaterial, right? So it could have a tissue construct, it could have partly things which are going to integrate with tissues and partly things which are going to be biocompatible. Similar with medical devices, which are built in certain cases with certain biomaterials and biocompatible materials, right, which are tissue compliant, right? So these extremely diverse set of equipments, devices, and tissues are handled as a part of clinical testing exercises, right? So in order to do that, the safety of medical device or a material uh, is the primary concern of any inventor, and for that case, even the soft tissue. So when I extract a piece of soft tissue from, let's say, cadaver, right, do I just take that piece of soft tissue from there and test it in my lab, put it in a mechanical test bench and test it? The answer to that is no, right? Because there are a lot of criterias which have to be followed as per the testing norms and ethical norms, right? So they have to be stored at a certain type of amount of temperature. They have to be enclosed just to make sure there is no infection or any kind of other contamination, right? They have to be stored and transported in a certain way, and whenever they are tested, you cannot just take a piece of tissue out of from a fridge and start testing it. It will have properties which are very different from the realistic tissues. So they have to be kind of conditioned and made sure that they come back to their real tissue property, which they would showcase like in a real human body or animal body. Then it can be tested. So there are a lot of rules and regulations and protocols which are to be followed, right? So, but at the end of the day, the primary concern becomes the safety and following the proper protocol, right? So sometimes as a whole, risk is, like counterbalance with safety sometimes. So you do not want to work on very, very risky projects involving soft tissues and other things. Like, for example, I do not want to extract a piece of tissue from a live human and test a few drugs, right? Just for the sake of it. Like, so that cannot be done. It's in extreme cases like where something is of a national priority, then safety and risk can be balanced that way. Otherwise, if it is very, very risky and the safety standards are low, the testing may not be allowed, right? Okay. So one of the key importance of clinical testing is to translate biomedical engineering innovations from lab to the real world applications. So that's one of the key purposes. So whenever we are testing tissues, be it from animal models or the human models, that should be with some purpose directed towards serving the society, and where we can eventually use this tissue testing for some greater good. Like, for example, like you're trying to develop this new type of skin graft. So if you want to test this with a real human tissue or a cadaveric human tissue, that is allowed. But if you just want to extract tissues and test them, that may not be allowed, right? So hopefully that makes sense. That has to have a kind of real application to it at the end, and it should be serving the greater good, right? So this should be possible only by providing the effectiveness in human subjects. So many of the tissue-based tests and applications which we have developed over time, and quite a few of them we have discussed during the course. In order to assess their effectiveness, sometimes we may say that we could use pig skin or cowhide skin, like to just uh, basically standardize human skin, right? That's not possible. You have to ultimately test it with cadaveric human skin or a real human skin in certain cases. Otherwise, you cannot say that this is a skin simulant which is similar to human skin, for example, or this is a skin graft which is going to behave very similarly in an actual burn victim as compared to what we have tested in the lab, right? So the effectiveness and everything which is human specific has to be tested on humans or cadavers, right? So that has to be done eventually. So that's one of the primary motto of clinical testing. From a safety standpoint, we have to look at biocompatibility when we are looking at tissue materials, like tissue engineered materials which may go inside the body, or there's a medical device which interacts with the body, or we are developing a skin graft which is an implant, for example, which needs to go inside the body. So we just cannot look at the mechanical properties and say this is good to go in the human body. That kind of conclusion is baseless, right? You have to just make sure that they're conforming with all the basic and generic safety standards. And one of the key criteria there is biocompatibility. We are going to define that eventually, but what this means is this particular thing compatible with the human body, right? Second is surface characteristics. This may or may not be useful for majority of the cases, uh, but you would find them useful if you're trying to devise skin simulants or test skin, for example, with textiles, interaction with sensors which are placed on the skin, and things like that, right? So that's where you're focused more towards the surface characteristics. Also, operator safety is important. So you do not want the operator to go there, extract 5 pieces of cadaveric skin which is infected, for example, right? That could compromise the operator's safety, right? So you want to basically go for cadavers or particular human models where the infections are minimal. It could be a case of trauma due to which the death has occurred, and those kind of cases are safe. So all these assessments are useful. So during COVID, if you're trying to do a test on a cadaver who uh, has met with death like due to COVID, like that's not ethical. or the right clinical test, right? So safety standards have to be maintained as the vertical number one. Second is sterilizability, right? You should be able to maintain a sterile environment, which is something which cannot be contaminated, or that contamination should not lead to some properties or outcomes which you do not directly predict in human bodies, right? So some of the key criterias and important parameters here are sterilizability should not be destroyed through something like autoclaving, for example, dry heating, radiation, ethylene oxide, which are common materials for fixing and things like that, right? So any kind of environment or chemicals or other additives which you may use to store your tissues properly or put them in the right order for testing should be in a sterile environment and should not be contaminated at any point, right? So this is the second vertical which is important. Third is manufacturability, right? So will you be able to, this is more to do with biomaterials and tissue engineering applications or medical devices, right? If you're trying to bring in skin simulants or develop tissue constructs, what is the strength, what is the durability? You should not just make properties which are cellular wise or at a much more micro scale similar in the durability with the skin, for example, right? It should have the strength of the skin, so the mechanical properties have to also comply with the real tissue properties, right? Next is it machinable? Like, can you use some sort of machines to mass produce certain things? Like, for example, skin graft, this is a machinable technology, right? You could use a machine and place a skin, the machine is known as a skin graft mesher. You can place a piece of skin, healthy skin, into it and basically mesh it, which basically projects a range of perforations on the skin surface, and basically that causes the skin to be expanded further more than what it can, right? And that allows it to expand further to aid in skin grafting applications and burn injury mitigation, right? So machinability, moldability in certain cases. If you're building a skin simulant or a biomaterial or a medical device, is this moldable with certain materials which you may have prepared using tissue constructs and things like that, right? So that manufacturability is also an important criteria, may or may not be useful in certain areas if you're just testing tissues, right? But otherwise, yes. Okay, so in clinical trials, you're basically trying to translate a research to get into the real subjects, possibly eventually. So here, evaluating the safety, efficacy, and performance is very important for the studies, right? So safety has to be maintained at all aspects, right? Like, an efficacy is how um, efficiency, how much is the efficiency of the overall system, what you have prepared. Like, if you're able to extract tissues, then how are you maintaining those tissues, how are you maintaining the properties and things like that? Are these tissues which you have extracted from a cadaver, for example, performing similar to how it would be if I took a live tissue out of a just freshly excised cadaver, right? Which is the death has happened like within the past 5 or 10 hours, right? Is it going to behave similarly? So that's a performance parameter, right? So in clinical trials or clinical tests, these three things are very important: safety, efficacy, and performance, right? In order to maintain all these aspects properly, we have to make sure that we optimize and refine our goals and sometimes the protocols in order to do them correctly. It could be that we are extremely good with the safety and efficacy, but performance wise, the tissue constructs which we are developing through bioprinting or some tissue regeneration techniques are not really performing the same way an actual tissue would perform at all, like in a clinical study. So that gives a metric of the performance. So you may have made something which is mechanically similar or biologically similar, but it may not show the right performance when you clinically test it. So that's very important, right? So these are some very important parameters of clinical testing which we have to keep in mind. So here, one of the key aspects which is highlighted here is the responsibility lies with the regulators, these are the regulatory bodies which ensure that the clinical tests are conforming to all the right practices to ensure that the safety and efficacy of medical devices, tissue constructs, biomaterials are all maintained rightly. Right? A wide variety of items are classified as medical devices, making medical device regulations pretty complex, right? So we have medical devices which are soft, we have medical devices which are hard. Soft example is a stent, right, which is going inside an artery. So you're basically trying to open a clogged artery using a soft device, right? So they go through a lot of testing already, but are they mechanically 100% compliant? The answer to that is no, right? So there have been few industrial tests which are currently done, but they may have to go through more tests to make sure that the stents do not fail at all. And clearly many of them are failing, right? There are nicer stents which stay on for a while, but there are stents which are undersized, oversized, or has undergone some thrombosis due to scratching the surface of the artery, and there is some tissue ingrowth. It's not able to maintain it, right? Like maintain the artery open, right? So there are so many other issues which can happen, but uh, these are all components of medical devices under soft and hard categories, right? So no standardized or universally accepted procedures are there for clinical testing to date. This is good to know, right? So majorly for medical devices, for tissues, it is much more protocol oriented, which have been kind of followed for at least I could safely say 20 years, 15 to 20 years, where how tissues have to be tested are properly protocoled, right? Like there are rules and regulations, but for medical devices, no, right? If you have a medical devices which goes inside the certain tissues inside the body, uh, it's very difficult to test it. You have to use the knowledge of finite element modeling, for example, how to standardize that. You have to, because you cannot test it experimentally. You have to basically test them using all kinds of loading mechanisms. Just a uniaxial tensile test may not work. You need to do uniaxial compression, maybe a multi-axial test. So all these accepted procedures when you're dealing with tissues and their interactions with other medical devices and biomaterials, it's not very clear as per norms, right? As per the clinical norms. So getting a little bit into the medical device, because this tends to define a category which often gets in touch with the soft tissues and the hard tissues, right? It's an instrument. It's an apparatus, basically, which implements, like a machine contraceptive implant in vitro reagent or other similar related article, including any component, part, or accessory, which is so anything of this sort, like instrument, apparatus, machine, contrivance, implant. These are all categorized as medical devices. So these could form components of such medical devices. For example, within an implant you could have 5 or 6 components, right, which are there. So they have to be overall recognized in the official national foundry or the US pharmacopedia or any other supplement to them. So there is a Indian pharmacopedia as well, so they have to kind of reflect somewhere as per the regulatory norms. So then the medical device has to be intended for use for diagnosis of the device or other conditions in the cure, for example, mitigation, treatment, or prevention of the disease in man or animals, right? So these are the standard terminologies which are used, standard terms and sentences. Intended to affect the structure or function of the body of man or animals. These are the two categories quite often emphasized, and which does not achieve its primary intended purpose through chemical action within or on the body of man or animals, right? So this is just a way things are written, like in the sense of like the function, right? So it should not do any sort of adverse chemical reactions or other things should not happen when a medical device or a tissue construct or a tissue engineered biomaterial goes inside the body, for example, right? So this is part of the broader clinical testing norms. Besides that, which is not dependent on being metabolized by the achievement of its primary, intended purpose. So no other adverse reactions, be it chemical, non-chemical, or any sort of tissue ingrowth and things like that, which are not desirable. That should not happen during these interactions, right? So that's what is emphasized here, right? So all medical devices, including an instrument, apparatus, appliance, implant, material, or other article, right, whether used alone or in combination, including a software or an accessory intended for by its manufacturer to be used specifically in human beings or animals, which does not achieve the primary intended action in or on human body or animals by any pharmacological or immunological or metabolic means, right, but which may assist in intended functions by such means for one or more of these specific purposes, right? So this is just a standard language which says that like so medical devices could have a very broad category which includes biomaterials, tissue constructs, tissue engineered patches, sensors, so many things which gets in touch with the human body, and they could be technically used for diagnosis, prevention, monitoring, treatment, or alleviation of any disease, so betterment of any disease or disorder, or any injury for that case, or disability, right? For example, if you're developing a prosthetic, it has to have the properties of the right hard and soft tissues, right? For example, the human extremity prosthesis, which is the foot prosthesis, which is a very common. There are transtibial, transfemoral, these kind of orthoses which are there. They're very standard with metallic parts. Can they not be built with soft and hard counterparts, like tissue counterparts? They can be, right? So that's where a lot of these clinical testings can be conducted in the future once these norms have been developed, right? So clinical testing also deals with investigation, replacement, or modification of support of the anatomy of the physiological processes, right? So if any of the medical devices, tissue constructs, biomaterials which goes inside the body creates any kind of replacements for the body. So this is clearly like skin replacements and other organ replacements, which we are all talking about during studying about bioprinting. That needs to be properly maintained in terms of protocol, like otherwise it cannot be allowed by the clinical testing norms. Supporting or sustaining life, so this should be supporting and sustaining life in general. This should not be something for just experiments and testing the positive or possible negative effects. This should all be for positive effects which benefits the patient or the human participant who is being used, right? So in cadaveric cases, that is very different. Like you could basically take out skin and other materials from the cadaver, but there are certain norms where if you take a huge amount of tissue from a cadaver, you have to stitch it back into the cadaver. This is as per of the general norms, right, which are there for cadaveric testing, right? Disinfection of medical devices or tissue constructs and other materials. This is also important. Control of conception, right? So these are all standard norms from the Ministry of Health and Family Welfare by the New Drugs and Medical Devices and Cosmetics Bill, which was basically released in 2022, right? Next is in vitro diagnostic devices. So this is typically these are some terms and terminologies we should all understand because these devices are going to interact with soft tissues, right? um, Directly or indirectly, and they may have components of tissue engineered materials, right? So in vitro diagnostic device is basically defined as a reagent or a reagent product or a calibrator or a control material or a kit or an instrument or an apparatus, equipment or a system, whether used alone or in combination thereof, intended to be used for examination and providing information for medical or diagnostic purpose by means of examination of specimens derived from the human bodies and animals, right? So in vitro diagnosis, long story short, deals with any kind of soft tissues or other kind of components, hard tissues which you've derived from the human body or animal models, and you're basically using these devices for all sorts of testing. So for example, a universal testing machine, if you're running a uniaxial test on soft tissues, then it doesn't necessarily have to be an in vitro diagnostic device, but that does come under one of these categories where you're trying to figure out the mechanical properties, for example, right? Okay, next is why not just preclinical testing? Why do we need a clinical testing in order to do everything which we want to do with soft tissues, right? So it ultimately depends on how the material is performing rather than how biocompatible it is, right? So these are some standard answers to a lot of FAQs which are there for clinical testing. Why not just preclinical testing? Because the idea is it is not good enough for a tissue material or a tissue replacement material to just be biocompatible within a system. Biocompatible here means like if I develop an artificial skin model in my lab using some sort of bioprinting and other tissue engineering protocols, can I just put that patch of skin on the human body? I do not know how it is going to react. It may be biocompatible, means it is compatible with the human body. It's not going to generate any adverse reaction, but it may not heal, for example, or it may not integrate with the normal tissue. That's a performance parameter. So even though tissues, materials, medical devices may be biocompatible, they may not have a good performance. And performance is more of a criteria which supersedes the biocompatibility aspect in a clinical testing. That's why preclinical testing is sometimes not good enough. Preclinical is when you're just testing for biocompatibility. Clinical is when you test it on human models or animal models. Now you would know whether this thing is going to integrate well in a mice model or a human model or things like that, right? So this kind of reality checks are very, very important. Okay, so biocompatibility, including the other preclinical testing, still are very adequate as measurable quantity, right? So many of the times you may find things to be biocompatible. It's very subjective. It could be biocompatible for 10 hours. It could be biocompatible for a week, for months. It may not be biocompatible due to some leaching and other chemical reactions and issues which may be caused after 2 months, right? You don't know. So biocompatibility is not a good performance parameter or not a measurable quantity to enforce usage of any kind of tissue engineered material or medical devices on the human body, right? So this has to be understood from a clinical testing standpoint. So, but the ultimate effectiveness can only be established through clinical trials. So like I said, this is very, very important. That's why clinical trials are important. So clinical trials largely depends on two factors: how the host responds to the material and how the material responds to the host, right? So these are some of the important aspects which you see, which is basically if I'm using a tissue engineered material or a medical devices on a human body, how the particular device or tissue construct is reacting to the human body and how the human body is reacting to this tissue construct. Both are equally important, and we devise performance parameters based on that to understand all these aspects, right? Further, so basically what happens is from getting back to the different levels of the clinical testing, you basically have the idea which needs to be validated in preclinical tests, and basically then you go for uh, a patient, right? You're looking to identify the need, design, materials, selection, synthesis, right? And you have this testing norms during that time: fabrication, device testing, and clinical testing, clinical use. So these are some of the common attributes which you think about when you are ideating on a particular problem, or what tissue constructs have to be used, or what kind of tissue materials have to be operated on, like in a surgical trial or things like that, right? So from a physical standpoint, you're looking at abrasive, adhesive, delamination, or corrosion based difficulties along with tensile strength and compressive strengths. From a biological standpoint, you're looking at absorption, enzymatic degradation, calcification, biocompatibility. So these are some of the things to consider where you would find further metrics to evaluate whether these are working fine or not, right? Makes sense? Okay, so let's formally define biocompatibility as per the ISO norms, right? These terms are going to come back again and again to us, so we need to understand these, right? So biocompatibility is the ability of a device material to perform with appropriate host response in a specific situation, right? So in a given situation where there is a surgery, or there is a particular device inclusion, medical device inclusion, or tissue construct inclusion, or a biomaterial inclusion within the human body, Biocompatibility is the ability of that construct or the device to perform appropriately how it should behave within that system, like a normal tissue. That's the answer, right? And have the right host response. So it should not create some inflammatory response where there's an inflammation around the implant, inflammation around the tissue. It's not adhering to the natural tissue. So things like that should not happen. So that's the idea of biocompatibility. If we say this particular tissue construct or tissue engineered material is biocompatible, that means it blends in well with the original tissue. Whether it heals or properly integrates mechanically in other things is a very separate problem, but it's compatible. It's not going to generate any adverse reactions. So that is what biocompatibility is as per as the DA's biocompatibility guidance on use of ISO 109931. So this is based on that handbook. Okay, so what are some of the screening tests for biocompatibility? There are several terms and terminologies which are often observed, which includes cytotoxicity, sensitization, irritation or irracutaneous reactivity, acute systematic toxicity, genotoxicity, right? Then we have further implantation effects, hemocompatibility, is to do with blood, chronic toxicity, whether this is leading to long-term toxicity, right? Carcinogenicity, whether any of this is carcinogenic, has possibility of causing cancer. Reproductive and development toxicity, whether if you have a tissue construct or a material, is it leaching some material or some chemicals within the system which may cause reproductive or developmental issues like in the later run. Biodegradation. So all these important aspects have to be considered while we develop any tissue constructs, tissue replacements, biomedical devices or biomaterials, right? So these are some of the standard clinical tests which are existing, right? So typically this is an example of you're looking at some of the medical device categories, and you're basically looking at biocompatibility test based category and contact duration, right? So this is again as per the ISO 10993 933 9931 category. So in this table what you're looking at, there are several types of devices or constructs or biomaterials which you will encounter in your day-to-day life products or medical devices and products. The first type is a surface device, right? That's a category. Surface device which only stays on the surface. So the contact example is intact skin, mucosal membrane, breached or compromised surface, or black path. No, that part is not there. So basically, surface devices includes a lot of sensors which are just lying on the skin. For example, you have a range of sensors which are in touch with the skin to measure, let's say, the sweat or perspiration in general, blood glucose, some other chemicals, biochemical markers, or something like that, right? So the typical contact durations are can be categorized as limited, which is below 24 hours, prolonged, which is 24 hours to 30 days, and permanent, which is more than 30 days, right? So if you have a pacemaker which is placed within the heart, right, like you are basically looking at a permanent device, of course, like it's going to stay for quite a while, right? So this very important matrix table basically tells you what are the some of the absolutely important tests and mandatory tests for each these kind of category of the devices. So for surface devices which are just on intact skin, so there is no skin breakage, there is no wounding, sensors and other devices which just sits on the skin, deals with a lot of variables. You are basically looking at three type of tests which are marked by cross there. You're looking at cytotoxicity, sensitization, and irritation or urocanous reactivity. These are only the three categories of tests which have to be followed, right? If you're looking at devices and tissue constructs, materials which are in touch with the mucosal membrane, uh, both ABCs are the three timelines, like the duration for which these devices are going to be in touch. You're looking at a range of tests, you know, besides the cytotoxicity, sensitization, and irritation test. You're also looking at acute systematic toxicity, material medicated pyrogenicity and surface subchronic toxicity. These are some of the terminologies which are used for the different tests, right? Clinical tests. Next is breached or compromised surface. For example, this is a tissue breach or tissue wound, right? For example, diabetic insoles, like which are in touch with probably a diabetic ulcer, right? So skin graft, which is in touch with a burnt skin, which is the skin is exposed, right? Or any kind of devices which are going inside the body, which are in touch with the tissue, right? On a breached surface, like not with a normal tissue, right? So they have to go through a range of tests which are given here, right? So X is corresponding to the, as per the ISO 10993 recommended endpoints for consideration, right? And the ones with O symbol are additional FDA, which is Food and Drug Administration of the US recommended endpoints for consideration. And many of the clinical testing norms in India and other countries are also adaptations of the FDA norms with certain relaxations based on the country. And few of the things may not be relaxed at all. They may be more stringent. Okay, so here what we are looking at is then the blood bath indirect, like external communicating. So if you have a device which has a needle kind of a system which is in touch with the blood flow and other vascular systems, you need a range of tests which are absolutely necessary and few mandated by the FDA, right? Then you have devices which are in contact with the tissue slash bone slash dentin, for example, dentin is of the teeth, or circulating blood. So they have a different set of requirements for the tests which are absolutely mandatory, right? So based on this table, you can basically categorize that if you're building a tissue construct which has to go, like within the body versus outside the body, or in a breach situation or a disease condition trying to treat with a biomaterial or things like that, what kind of clinical tests, test categories which you have to be mindful of, right? So that's what this table tells you. Then comes the implant devices, which includes tissues or bone interacts, right, or interactions with the blood, right? They have a range of, again, compliances which have to be followed, right? So a range of tests have to be conducted besides mechanical tests. Mechanical is not part of a direct clinical requirement, but you have to do a bunch of clinical tests along with the mechanical tests to make sure a tissue actually is able to be tested in the way that it helps somewhere in the overall human benefit, right? Like, so that has to be looked upon. Okay, so the next is we want to look at very quickly into the classification of medical devices, because this is an implication of how they interact with the soft tissues, right? So the classification of medical devices are based on anticipated risk while using the device. So very simple criteria: what is the level of risk while using the device? So that's the basis of classification. So they are basically classified as class A, which is low risk, for example, thermometer, right? Or tongue depressor. These are some examples. So that's a class A low risk device, right? Next is class B, which is a low moderate risk device such as suction equipment, hypodermic needle, and things like that, right? Then class C is moderate to high risk, which includes ventilators, bone fixation plates. These are some examples. Class D is extremely high risk, like heart valves, AICDs, and other biomaterials and other techniques, other devices which are there in touch, right? This could have very complex tissue constructs also which goes inside the body, right? So this is the broad class of medical device classification, and many of the tissue engineered based biomaterials and other technologies also fit in these categories based on their risk levels, right? So they are basically also kind of looked upon from a need versus necessity standpoint for clinical testing. So similar to preclinical testing, not all medical devices need to go through all stages of clinical trial, right? This is very important to know. So this depends totally on the classification of the medical device or the biomaterial or the soft tissue mimic, right? So it depends on the ability of existing data to adequately address the benefits risk profile claims and side effects of a medical device. If you do not have any data and you're trying to build up a skin graft which you want to claim that this is going to work on burn patients, that is not going to work, right? Besides mechanical tests, there have to be a range of tests which have been tabulated in the previous slides, and if once they're followed, the range of data has to be presented in order to do an overall risk assessment, and then based on that, of course, the necessity of a skin graft, improvised skin graft, is fairly high, so that is pretty intuitive. But if the need and the data doesn't support it, then that clinical testing may not be conducted, right? So these kind of aspects have to be looked upon while planning clinical tests. So mostly the implantable devices like the class 3, 7, class 4 need to go through all stages of clinical trials. So if your tissue construct, if your biomaterial, if your tissue mimicking, tissue regeneration or replacement material, be it prosthetic, orthotic or whatever, falls in any of these class 3 or 4, you need to go through an entire stage of clinical trial in order to possibly use them in humans at some point, right? The ultimate decision lies with the legal authorities, and there could be certain other things and medical bodies which are involved and have experience dealing with these things, right? Okay, so clinical investigation, to define it, so we have talked about clinical testing for a while, right? What is preclinical testing, what is biocompatibility? We have learned about a few things and why clinical testing is important. That's hopefully clear. The next part is clinical investigation. We just want to define this. It's a systematic investigation or study in one or more human subjects, right? So this is the definition, undertaken to assess the safety, clinical performance, and or effectiveness of the product, right? So it cannot be done only on one human subject. It has to be done on a range of human subjects. They have to have a declared statistically relevant, demographics, like, so it could be a certain disease population in which we are trying to test a few medical devices and biomaterials, versus like it could be healthy subjects where we are trying to look at some replacements, surgical replacements or things like that, right? So this has to be done, all these processes, with the idea to assess the safety, clinical performance, and or the effectiveness of the product. So that's what a clinical investigation is, right? Let's go further. Why do a clinical investigation? This is to ensure that patient safety is maintained by rigorously assessing the potential risk and benefit of new interventions, right? Be it so that you are going to face all of these if you're trying to work in the area of biomaterials or uh, tissue engineering problems, which where you are going to eventually engineer in tissue, and basically if you have this tissue construct which you're going to replace a particular tissue on the human body, you have to go through all these steps, and that's why understanding the clinical testing norms is important, right? So potential risks and benefits of new intervention is a must. Patient safety with respect to that, clinical testing should help optimize the performance and usability of biomedical innovations, right? So tissue engineering, for example, again is a biomedical innovation. So the clinical testing should not only just validate that this is a great piece of tissue mimic which you have created, for example, it should be able to improvise the performance of this tissue mimic through better adhesion with the body, better healing, and things like that, right? Clinical testing should also serve as a bridge between academia, industry, and healthcare institutions, right? So through this proper tests only, we'll be able to address the problems holistically and through approvals of the academia, industry, and the healthcare institutions, right? Otherwise, we're just working in silos and working on some problems which may not be for greater good when it goes out. Collaboration between researchers, engineers, clinicians, and regulatory bodies is important for successful transition of biomedical engineering innovations into clinical practice, right? Um, Clinical testing also helps in gaining practicality and acceptance of new devices. So this is again a very important aspect, right? Okay, so there are two major types of products when we talk about clinical testing. The first category is pharmaceutical products, which is basically to do with all kinds of medical drugs, And then you have medical devices, which is a broader term for all sort of biomaterials, tissue constructs, hard implants and other devices which goes inside the body, right? So basically, if we talk about from a vertical standpoint, the type of organizations which brings products to the market. So pharmaceutical products are majorly put to the market through large and established pharmaceutical companies, which we are aware of, like for example, Sun Pharma, or you have let's say Leica Labs. Like there are so many, Dr. Reddy's Lab and things like that, right? Uh, for medical devices, it could be highly variable. Many startups and small medium enterprises, as well as large medical technologies companies, may be involved. Even research institutes may be involved, right? So there's a quite striking difference between pharmaceutical and medical device based products. Time when clinical evidence is generated. Based on that, generally for pharmaceutical products, you're looking at pre-market clinical evidence that this particular medical drug is effective and properly treating a certain disease, right? But in terms of medical devices, you're looking at both pre and post market studies, right? So it could be that you're trying to look for a pretty long time study of 10 years to just observe how the patients behave with a certain type of bioprinted skin graft, for example. So this could be one of the possibilities where you do some pre-testing on a certain animal models, human models, and go into the market, and then you want to keep on testing it for a while to look at the efficacy, safety, and other parameters. The clinical development phases for pharmaceutical products are highly standardized by now, so they have a clear set of phases 1 to 4. Medical devices, however, are less standardized and are very product dependent, right? So they may use different kinds of manufacturing and non-manufacturing processes for the same. Next is clinical study design. For a pharmaceutical product, it's highly standardized. Double-blind randomized controlled trials are expected, and for medical device, less standardized pivotal trial, often done after the initial marketing is initial marking, sorry, is commonly observed, right? Irreversible effect on study subjects, which can be caused from these kind of products, includes pharmaceutical products. They're very rare because they're tested on a range of uh, human subjects and a larger population. Medical devices, it's very common to have irreversible or traumatic effects on the human body, particularly with permanent implants. So think about and completely untested bioprinted skin based scaffold printed put on a person with severe burn injury, and let's say this has caused permanent damage, right? So this is again under the medical device category, which we have to be careful of. Okay, so if we talk about regulations in India, we are looking at, this is all from public literature and public information, try to extract a few pointers here. So Chapter 7 of Medical Devices Act 2017 deals with the regulations of clinical investigation of medical devices and clinical performance evaluation of new and in vitro diagnostic medical devices, right? So this is what the act is. Permissions needs to be taken from the central licensing authority, right? Permission needs to be also taken from pilot investigation or for both pivotal investigations. These are two types of investigations which are carried forward. Medical devices claiming substantial equivalence to predicate devices shall not be marketed before getting the approval. So many of the devices which are already existing, devices which are saying that it is quite equivalent to what the market has produced, sometimes to bypass the rules of clinical testing, they will not get approval that easily. So that's as per the norms of the Indian government, right? So ideal D framework for implantable device. This is another very commonly used framework which talks about the ideal is coming like this: idea, development, exploration, assessment, and long-term study. These are all very, very important aspects of clinical testing. These are typically introduced for developing and evaluating new surgical procedures, right? Uh, Broadly divided into 4 stages. You're basically looking at stage 0, which is preclinical, stage 1, which is first in human, stage 2, which is prospective developmental studies, stage 3, randomized controlled trials or alternatives, stage 4, long-term studies, right? So this is as per the ideal framework, and this can be looked upon into this particular literature from IDLD, a rational framework for evaluating and regulating the use of medical devices. This is in BMG, Clinical Research Education, right? So this is quite popular. So what are these stages particularly where we should understand, because this will all be part of all our testing and other research activities or industrial research activities which we conduct in the future, right? So stage 0, which is the preclinical stage, includes the product design, materials, and functional components are determined and tested to prepare the device for first in human studies. So here you're looking at mechanical tests, all kinds of mechanics testing, biocompatibility testing, and all the tests which are kind of laid out in that table which we have discussed a while ago, right? Animal models are often used to optimize new techniques before human studies begin. So if you have developed a new graft or a new implant, which is tissue based, for example, you would like to first of all implant it on some animal models to look at some of the trends, and then if you see some nice healing trends or nice levels of like reorganization of the tissue and things like that, you can then potentially test it with enough data on human subjects, right? Typically, you do not go for humans directly. So the principles for stage 0 reporting will have to balance the need to protect intellectual property and prove that appropriate ex vivo tests of safety and reliability have been conducted, right? So this is often the stage at which a publication may come out, or there could be some intellectual property associated with the work where you have had a bunch of tests which are conducted to assess the safety and reliability of a certain device or tissue construct, for example, right? Okay, stage 1 is first in human. So for any kind of first in human studies, you need a universal registration of studies which could only allow the identification of benefit and harm themes and trends by surveillance of incoming reports and detect possible device harms at an early stage, right? Ensuring that unsuccessful innovation is not repeated through ignorance. What this means is there is a proper universal registration of studies which is there, and in this registry they can always tally when a new proposal comes for clinical trial first in human studies phase 1, right? That has any of such similar studies ever been conducted in the past, let's say 40-50 years, or entire timeline where there has been massive catastrophic failures or harm being caused to the humans? If that is the case, then these kind of studies will be totally neglected and not allowed to be carried out, right? So this is one of the first steps and criterias in the first in human phase 1 studies, right? Innovators or principal investigators should be ethically obliged to search the registry. So you can find a lot of public information in the registry before embarking on a first in human study to avoid repeating a harmful error reported by another investigator. So many a times, like if you're working as a part of the industry or a researcher, you're working on a medical device, biomaterial, or a soft tissue implant, for example, or a soft tissue construct, you want to place it on the humans to look at the effects, but let's say such a study has failed miserably. So let's say you have created an artificial liver, somebody was able to transplant that liver, may do some animal studies, and in the human studies there was a death reported, for example, of 2 people, for example, right? You would not want to embark in that study line unless you have extreme amount of evidence and collaborative evidence that the study is going to work or make it through, right? So these are some very important aspects and responsibilities of the principal investigators, right? But how to protect reports of device innovations that proved harmful for being used for legal challenges, but right? So many a times it could be that device innovations led to harmful issues, like, but and there could be legal challenges. So sometimes it may not be public information. So that is where the registry can look at it and come back to you, right? A preventive measure is to spend a good time in stage 0 and conduct vigorous and rigorous preclinical trials. So nobody should ever rush into conducting clinical tests on the studies like which are on human subjects directly, right? There should be a lot of mechanical studies, mechanics studies, biocompatibility, biological, biochemical studies before it moves to the possibility of animal tests and further to human tests, right? And that is what we do as a part of all the research aspects, and that is what needs to be complied ethically. Stage 2 is prospective developmental studies, where mostly technical device iterations occurring during the stage 0 to 1 and involve a single manufacturer, right? So let's say I'm using a single manufacturer for making my skin graft. I'm using this as a central example just to kind of not have multiple examples clutter your minds, but skin graft, which we are talking about for a while, and I've showcased some of the examples earlier. So let's say there's this particular device manufacturing company which can now process all the skin samples to create the right type of skin graft. They may have gone through a set of iterations to get the crafting done properly, right, or the meshing done properly. Might be one device which is not projecting the slit patterns properly. So after all these iterations, you need to go for the stage 2 once they have been tested and tried. There needs to be consensus among the medical professionals that there is no issue with medical device or any kind of material at this stage. Is stage 2 aims through prospective exploration studies at facilitating progression to a definitive randomized controlled trials in stage 3. The stage 2 kind of makes its way for stage 3 if done right? So stage 2 can be basically considered as an extended pilot study, right? Uh, it also requires that there is a considerable enhancement in the average level of the clinical evidence, right? Supporting new devices would be set up such as for a definitive study. So basically this is making path for a definitive study. So you should uh, conduct stage 2 in a wide range of population in order to make sure that there is absolutely no harm and things are actually pretty effective, like in terms of whatever the outcome measures we have decided for this particular material or tissue construct or medical device, right? And based on that, the test stage 2 can be conducted. Further, uh, we should talk about briefly into the pilot investigation and pivotal investigation, how they are different. In a pilot study, you're looking at clinical investigation to be carried out for the first time in human, right? Like this is a pilot clinical test, phase 1. After data analysis is a from pilot study, you get a pivotal study result, right? So both are very, very different. Pilot is again exploratory in nature, when pivotal is more of a definitive and confirmatory approach, right? So both studies are very, very different. One is more of a hands-on and realistic, and other one is more, theoretical. You basically have in a pilot study a smaller number of participants, and pivotal has a large number of participants. I'll give you an example. For example, like I want to test the efficacy of diabetic insoles in a lot of participants, right? So I've created this new type of diabetic insole. I want to test it on like 10 participants or 20 participants. That's a pilot study. There are 20 types of diabetic insoles in the market. This is an example, and I'm trying to test the efficacy of all these diabetic insoles. That's a pivotal study. I'm gaining a lot of data from the market and from a larger population, and I'm putting together the work. So it's already a defined norm that diabetic insoles are there in the market and popular. So I'm trying to get a larger feel of things, right? In a pilot study, you're also focused on assessing the feasibility, eligibility, potential harm, and validating outcome method, right? And pivotal studies, your overall goal is to assess the safety and efficacy, right? So this is a nice table to look at from a pre-market and post-market standpoint. You're looking at a clinical stage, and the descriptions and types. In preclinical, you're basically looking at an exploratory type, which includes in vitro and in vivo studies. Like in vivo are animal-based models and a lot of those kind of studies. In vivo can be human too, but like more often it's animal. Then you have pilot studies followed by that, which is exploratory and confirmatory. Exploratory is when you have certain hypothesis that this may react in such a way, right? This particular tissue engineered material may work, right? That's an exploratory, and this is going to work. I'm just going to confirm it through different tests. That's a confirmatory. That's a pilot study. So this is typically the first in human pilot study, exploratory study. This would have like feasibility studies, proof of concept. This is typically an investigator initiated, funded by some government grants or private grants, right? Then you have pivotal study, which includes confirmatory studies like pre-market, phase 3 kind of studies. Sometimes you have post-market, investigator-initiated PMCF studies and post-approval studies as well. So these are majorly interventional, right? This is the burden to the human body. Preclinical are mostly non-interventional, like they do not directly involve the human. This right? So that's exploratory in nature when you use a lot of animal models and things like that. So that has no burden on the humans. Then you have another one which is of a post-market surveillance study type, is an observational study. So these are the different categories in which you will see a lot of literature on any area of soft tissues or medical devices, for example. Observational studies include post-market effects of CIs, like medical devices, PMCF studies, investigator-initiated. These are registry-based studies, surveys, case series, cohort studies, post-market surveillance studies, and all of these are highly non-interventional. Okay. So with this discussion, we are going to continue from here and talk more about the clinical tests and dive into some very interesting case studies which uses human tissues for mechanical tests also. So we'll get into the specific tests in the upcoming lectures. So just stay tuned. Thank you.